Hello, thanks for tuning into this presentation. I'm going to be discussing our research on the impacts of 3 types of anthropogenic den disturbance on female black bear movements and impacts on cub survival. My name is Darcy Doran-Myers. I'm with the Florida Fish and Wildlife Conservation Commission at the Research Institute in Gainesville, Florida, and I am the Assistant Black Bear Research Coordinator for the state. We research bears throughout Florida in order to inform our science-based management of Florida black bears. We have 7 geographically and genetically distinct subpopulations of bears in the state, shown here in dark brown. The research that I'm going to be sharing today was conducted in the Apalachicola subpopulation in the panhandle of Florida, which is circled in red. Denning chronology and ecology in Florida may be different than some other research or study areas. So first, I'm going to give some background on denning bears in Florida. Florida has mild winters. It rarely gets below freezing, and bear food is available year-round. Only parturient female bears consistently den. So for this research, we focused on parturient female bears only. Most dens are open nests on the ground. Very few are excavated into the earth or are in tree dens. Our parturient female bears den from the 28th of December to the 14th of April on average, which is 107 days, but there's a big range across individuals from 70 days to 146 days. We also observed that parturient bears often had multiple den sites per winter. The mean was 2.4 den clusters per female per winter, but it ranged from 1 to 5 den clusters per winter. When I went to read the literature on individual scale responses to den disturbance, I found that the bulk of research is 20 or more years old. Linnell et al. 2000 wrote a really great meta-analysis on den disturbance in black bears, brown bears, and polar bears. For black bears, they found that den and cub abandonments were caused by human disturbance. I also read the existing accounts of denning bear responses to prescribed fire, the most notable of which was Land 1994, which observed an unconfirmed cub loss as a result of prescribed fire. Overall, there has not been a lot of recent research on individual level responses to den disturbance within the past 20 years, and there have been very few studies that have employed the use of GPS collars to do this type of research. For any type of den disturbance, I did not find an estimate of how far females flee from those disturbances, how long they are away from their dens, how long it takes them to set up a new den after a disturbance, or how far away their new dens are from their disturbed den sites. In the Apalachicola subpopulation, we trapped in GPS-collared female bears as part of a demographic study, the summers of 2016, 2017, and 2018. In the springs of 2017, 2018, and 2019, we located the natal dens of those collared bears. During the denning period, we monitored female bears for their responses to 3 types of den disturbance. The first was a mild human disturbance, which we refer to as a den creep. The second was an intensive human disturbance involving cub capture. And the third was prescribed fire. To monitor female bear movements, we use their GPS locations provided at 2-hour fixes. We conducted den creeps in mid to late February after most cubs were born. Den creeps involved quietly inching closer to dens to confirm female reproduction, close enough to sometimes see the female bear, but more often to hear the cubs squawking or nursing. That is the sound of a cub nursing that we would be listening for. Typically, we would get about 20 to 30 meters from the den, which is a distance from which the female could presumably hear, see, or smell us. Usually we conducted 1 den creep per den, sometimes we would do 2. We conducted cub capture events in mid-March. We would purposefully startle females away from her den and her cubs. We did not use any immobilization techniques. We would pick up the cubs, carry them about 50 meters from the den site, work them up, and return them within 30 to 60 minutes. We usually conducted 1 cub capture per den. Sometimes we would conduct 2. The last disturbance type was prescribed fire. Florida manages many of our public lands through frequent winter burning In Tate's Hell State Forest, where most, around 80% of our bear dens were located, burns occurred from January through April. We used post-burn evaluation records to associate bear movements with a prescribed burn conducted nearby. We also looked at USGS satellite images taken before and after the burns to find the distance from the den site to the nearest burn edge. And for some that were closer burns, we also measured the distance on the ground. So now we can discuss our results. We conducted 44 den creeps across 3 denning seasons. We observed one bear fleeing a short distance, about 40 meters, and then returning quickly to her den. No bears moved their den sites, and no bears permanently exited their den as a result of a den creep. Overall, there was little noticeable effect of den creeps on denning bear movements. We then conducted 42 cub captures and observed a very different response to this disturbance than we saw for cub creeps. All denning bears fled this type of disturbance. The distances the female fled from the cub captures are shown in the violin plot on the right. They fled an average of about 400 meters from their den site. Most distances were below 500 meters, but there was a big range, and the maximum distance was almost 1.9 kilometers. And here we have plotted time in hours away from the den after cub captures, which was variable as well. The females returned on average 6 to 9 hours after the disturbance, and there were a couple outliers above 20 hours. The maximum time away was 50 hours or over 2 days. After returning to the den after the cub capture disturbance, only 18% of bears re-denned at the same den site that's shown in yellow. Most bears, about 71%, moved their dens to a new site, which is shown here in orange. And 11% did not re-den and instead began post-den emergency activities, so essentially exited their dens early, and that's in red. Of those bears that re-denned in a new den site, the new den site was on average 127 meters from the disturbed den site, and it took the female bears about 2.5 days to establish the new den site from the disturbance. We also observed one case of cub abandonment as a result of cub captures. A female bear fled the cub capture disturbance and she did not return or move back in the direction of her den for about a week. So we determined that she had abandoned her single male cub. We were then able to intervene. We picked up the cub, we got a veterinary care. His name is now Maximus, and he lives at a zoo run by Florida Parks. The last disturbance type was prescribed fire. Data were available for the 35 dens located on Tate's Hell State Forest. Of those 35 dens, 11 or 31% were located within 1 kilometer of a prescribed fire. The average distance from the burn edge to the den site was about 270 meters, but there was a big range from 0 meters, meaning it burned through the den, to 950 meters, or almost a kilometer, but we observed reactions to fire as far as 680 meters away. For one of those 11 dens, we did not have any response data, so our sample size for responses is only 10. 60% of bears fled their den on the day of the prescribed burn, an average of 160 meters, but up to almost 800 meters. These bears returned between 7 and 11 hours on average, but that ranged up to 22 hours. 7 out of the 10 bears in yellow stayed at the same den after the disturbance. 2 out of 10 moved their dens to a new den site, and 1 bear lost her litter of 3 cubs, and she did not re-den after the burn. Those 2 that re-denned in a new den cluster moved their dens only a short distance, about 24 meters, and they did so within 0.3 days or about 7 hours. The 1 bear that exited her den and did not return after a prescribed fire lost her litter of 3 to the prescribed fire that occurred in April. The photo on the left shows the den depression where the female and cub sat, which was burned through. We were unable to recover the carcasses of the cubs. We recovered 2 of the 3 cub collars that were slipped and partly burned. And we saw that the female's large movements after the fire indicated that she was moving without her cubs. In summary, we found little effect on parturient denning bear behavior due to den creeps. Most bears did not noticeably react to our quiet but close presence. We also found variable effects on bear behavior due to cub captures, where some individuals' reactions were minimal and some were extreme. Most bears moved their den to a new site directly after the disturbance, and we observed 1 instance of cub abandonment due to a cub capture. We lastly found variable effects on bear behavior due to prescribed fire, where some individuals did not noticeably react and other reactions were extreme. We observed 1 instance of litter loss due to prescribed fire. We conducted this study because in order to effectively manage bears that live in disturbed landscapes, it is important to understand their responses to some increasingly common anthropogenic disturbances. And as researchers, it is also essential to understand their reactions to our research activities in order to responsibly conduct our research. The results from the study are applicable to those that are conducting research on bears that den. It's also applicable to land managers that are seeking to understand the impacts of winter prescribed burns on wildlife, or to understand the impacts of human activities which may bring people close to denning bears. I have included my email on this slide if you would like to chat with me about this research. Thanks all for your time and your interest in den disturbance, and I look forward to hearing your comments and your questions.
Hi, everybody. Thanks for attending my presentation today. My name is Nick Fowler, and today I'm going to be speaking about a manuscript which was published in Mammal Review this year, titled Denning, Metabolic Suppression, and the Realization of Ecological Opportunities in Ursidae. So denning is a complex and diverse behavior exhibited throughout the Ursidae family, though terminology has been somewhat inconsistently applied. Classically, maternal denning has been used in reference to parturient females in all species and is characterized by seclusion, reduced activity, and degrees of fasting during cub production and neonatal care. What has loosely been termed facultative denning represents denning behavior by non-parturient bears in response to reduced seasonal food availability, with the denning period coinciding with the period of cub production by parturient females, and the den site simultaneously performing both maternal and facultative functions. Additionally, among and within species, there appears to be high variability by geographic area and across sex age groups, and this appears to be driven by extrinsic and intrinsic factors influencing seasonal energy budgets. This includes observations that some sex age groups may reduce or forego denning altogether in some circumstances, and this blurs our definitions of what we generally collectively refer to as denning. When this is considered across the family, it's made more complicated by a lack of research on maternal denning behavior in our least studied species. So, as we describe in our review, this requires us to first define 3 distinct types of denning behavior. Maternal denning, being in reference to the period of seclusion undergone during cub production and neonatal care, which is not generally associated with reduced food availability or metabolic suppression. Next, we can distinguish between metabolic and metabolic maternal denning, where there is substantive evidence of metabolic suppression generally associated with a period of reduced food availability and/or prolonged fasting. Now, Ursidae phylogenies have received a lot of attention, and they've been revised a number of times. This has placed the trait of metabolic and metabolic maternal denning as monophyletic and as paraphyletic, though a common ancestor or mechanism regarding the lack of expression of this trait has not been really fully explored. This was until Wong et al.'s 2017 identification and placement of Protarctos abstrusus, whose dentition, habitat, and paleoecology indicates a basal derivation of metabolic dinning to both the Tremarctinae and Ursini groups. Of our extant species, this includes the Andean bear, Asiatic black bear, sun bear, sloth bear, American black bear, brown bear, and polar bear. On the figure on the screen, Wong et al. 2017 characterized species by evidence of what they broadly termed hibernation, and this supports the paraphyletic retention of this trait, denoted by the sleeping bear symbols. So in Ursidae, weight loss during dinning or fasting occurs proportional to duration and directly influences body condition, which is a common index of fitness. Exhibiting metabolic suppression during dinning, whether it be in response to reduced food availability or for the purposes of cub production and neonatal care, would therefore offer fitness-related benefits when extrapolated to evolutionary time. It's widely accepted that the Ursidae family is the product of one or more rapid speciation or adaptive radiation events. However, a mechanism driving these radiations has not been clear. Yoder et al. 2010 identified 3 sources of ecological opportunity promoting adaptive radiations: the adaptation of key innovations, the colonization of new habitats, and the extinction of antagonists which serve to release lineages from natural selection. We hypothesize that in Ursidae lineages, the realization of such ecological opportunities has relaxed selective pressures on seasonal energy deficits, contributing to the interspecific variability in den behavior. To evaluate our hypotheses, we searched the ecological, phylogenetic, and paleontological literature to attempt to first characterize and identify an origin of types of denning behavior, second, to trace the retention of this trait through lineages, and third, to identify sources of realized ecological opportunity which may have resulted in relaxed pressures on seasonal energy budgets. We contextualize our results in reference to extant species ecology. Through the results of our review, we present a revised species relationship figure displaying the expression of metabolic denning in Ursidae. This figure was redrawn from Krauss et al. 2008 with refined phylogenies from Kuchera et al. 2014, which was also used by Wong et al. 2017 for placement of Protarctos abstrusus. Species which reportedly exhibited metabolic denning are bolded, and gray horizontal bars are the approximate temporal spans in the fossil record. As there's not time to fully unpack our review, we're going to focus on a few aspects regarding ecological opportunities which contributed to the expression of metabolic den behavior and likely the adaptive radiations in Ursidae. First, the Andean bear, or Tremarctos ornatus, our second most distantly derived lineage and only extant member of the Tremarctinae. So the Florida cave bear, Tremarctos floridanus, is a suspected ancestor or sister species which evolved in eastern North America. This species reportedly exhibited metabolic denning, and its distribution was presumably restricted by the Borophagus or gluttonous-eating wolves and the Agriotherium bears of western North America. It's theorized that with extinction of these antagonists, along with the immigration of bison and tuquus into North America, Tremarctos floridanus expanded westward and eventually southward into South America through the Great American Biotic Interchange, along with the Arctotherium species or the South American short-faced bears. Through colonization of new habitat, these lineages entered an environment devoid of competing scavengers and quickly proliferated into a variety of species and/or subspecies, filling this available niche space. Soybelzon et al. 2009 reports fossil evidence of an adult female Arctotherium and dependent one-year-old offspring located in a cave suspected to be used for the purposes of metabolic denning. Now, this indicates preservation of metabolic denning behavior into South America, though it is not known how long this trait persisted in Tremarctinae lineages. Presumably, to avoid competition with other now extinct Ursidae species, the Andean bear colonized the cloud forest regions of the Andes Mountains and adapted an omnivorous to hypocarnivorous diet, utilizing year-round available forage, primarily in the form of Bromeliaceae. The species also adapted a smaller body size and longer forelimbs than hind limbs, promoting this more arboreal and high elevation lifestyle, unlike the Arctotherium species, which were more suited for scavenging and cursorial locomotion. The realization of these ecological opportunities reduced the selective pressures on seasonal energy deficits, and in our summation resulted in the cessation of metabolic denning behavior. When considered broadly, these same factors also likely contributed to the observed rapid radiation of now extinct Ursidae species in South America. Within our review, we subsequently worked through each of the 8 extant bear species lineages in a similar fashion from the Old World Ursids and back to the New World and our more recently derived lineages. Next, we characterized the denning ecology of the 8 extant species, providing comprehensive review of the expression of metabolic maternal, and maternal denning behavior, as well as the factors which contribute to these observed expressions. We highlight how both among and within species, the colonization of new habitats, key innovations, and antagonist extinctions have relaxed selective pressures on seasonal energy budgets, which is reflected in den chronology and fasting behavior. We also demonstrate how this is consistent with the variability and reproductive ecology with energetic and fitness-related consequences. Lastly, we incorporate the author's collective experience in the mobilization of several hundred American black bears in winter dens spanning 31 degrees of latitude. We suggest a continuum of metabolic den behavior across and within species, ranging from short-term fasting and seclusion resembling prolonged slow-wave sleep to the pronounced metabolic suppression strategies of boreal ursids, which may exceed 180 days annually. In conclusion, our review provides a plausible mechanism for the observed adaptive radiations which typify the Ursidae phylogenies. Though these phylogenies are often revised, our study is largely irrespective of topology and should withstand any changes in species position. Our review also supports Wong et al.'s 2017 placement of metabolic denning as basal to Tremarctinae and Ursidae species. However, when considered along with Spady et al.'s 2007 review of the evolution of reproductive seasonality in Ursidae, we suspect further research may support a pre-Alleropodinae derivation of metabolic denning behavior. Additionally, with further research, we expect the eventual use of terminology measuring the depth of metabolic suppression rather than discrete classifications. Selective pressures are the drivers of natural selection, and understanding species plasticity to these pressures improves our understanding of evolution and may also have practical applications to predicting species responses to novel selection pressures in the future.
Hi everyone, I'm going to be talking about declining salmon use by coastal brown bears of Katmai National Park and the ecological impacts of that change. This work was completed during my PhD while I was working at Washington State University. Katmai National Park is located in southwest Alaska at the start of the Alaska Peninsula, and it's best known for Brooks Camp, where bears congregate at Brooks Falls to catch salmon like in this photo. However, Brooks Camp is located on the interior of the park, and the park is also home to an impressive 497 miles of coastline, where there are abundant and equally charismatic brown bears. Bears on the coast live in close association with the marine environment. They use wide open coastal tide flats and nearshore islands, intertidal and inland streams, and tidally influenced salt marshes, in addition to other inland and terrestrial resources. They rely not just on salmon, but on salt marsh vegetation, clams, and marine mammals in those coastal areas. Because their lives and the resources they use are so closely tied to the oceans, bears allow us to study the effects of ocean changes from land, which can often be much easier than working in the ocean itself. From land, we can look at how things like rising water temperatures, changes in pH, oil spills, increasing vessel or human traffic might affect marine organisms, bears, and the marine terrestrial interface more broadly. We chose to examine bear diet and habitat use as a lens for looking at ocean change. Previous research in the area allowed us to compare present-day bear habits to those of bears in the 1980s and 1990s. We collared 31 adult female bears, or about 10 bears per year, between 2015 and 2017. We collected blood and hair for stable isotope analysis of the diet. And we supplemented those data with direct video and photographic observations. We used carbon and nitrogen stable isotopes, foods that we had observed bears eating, bear blood and hair, and the isotopic mixing model MixSire to estimate seasonal and annual diets of bears. Diets in the early part of the year before salmon arrived, so May through early July, were about 99% plant matter. Diets in the later part of the summer after salmon began running were 73% salmon and 27% plant matter. When we looked at the annual diet, we found that bears consumed 28% salmon and 72% plant matter. When we compared these results to data from 1989, we saw a drastic difference in the estimated annual diet between the years. Back in 1989, we saw only 62% salmon and 31% plant matter. So bears used to consume almost twice as much salmon and half as much plant matter as they do now. This decrease was pretty surprising to us, so we decided to look for further evidence of these changes that might have taken place through time. When we looked at observational data on the number of individual bears that were observed at one of the primary bear congregation areas on the coast, we saw that annually the number of individuals had decreased by 64% between a previous study in 1999 and our study in 2016. On a seasonal basis, the decreases were most pronounced during the salmon season, with a decrease of 78% in the number of bears we observed during our study. We investigated this salmon season link just a little further and looked at aerial survey data for chum and pink salmon, which are the primary forage species for bears on the coast. And those data showed that during previous studies shown here with the red arrows, that salmon abundance was high, but that during our study, shown by the blue arrow, the number of salmon had decreased by about half. Other research, such as work by Malik and Cox, also looked at pink and chum salmon abundance and found that there had been a shift in abundance through time, with recent productivity of salmon stocks in the Katmai area being substantially below average beginning around the 1990s. Our results showing a significant change in bear diets to a lesser use of salmon resources, accompanied by reductions in the number of bears seen during salmon availability, and observed decreases in salmon abundance, suggest that changes in the ocean are impacting how bears feed and use space. Of course, it's true that other things have changed during this time, and one of those things is a drastic change in human visitation in the area. Human visitation has increased 1,000% over 17 years. And that could have some effect on what we're seeing in the bears. There's also some question as to whether the change we observed in salmon abundance and bear diet are part of some long-term directional change, or whether they were a result of a more ephemeral change like the warm water ocean event, or the blob, that took place during 2013 to 2016, which was during our study. Regardless, we think that what we observed is representative of significant changes that can and will take place as the ocean warms and other things within the ocean change, whether that's a continuation of something long-term that's already happening, or if it's a little further out into the future. It's likely that the changes we saw are a signature of what may be to come. This study was also a lesson in not assuming that conditions observed in one area are representative of conditions in other areas that we might manage, even if they're in close geographic proximity. So back in Brooks Camp, which is still a part of Katmai National Park, the bears there are making national and international news for how fat and productive they are in the phenomenon that is Fat Bear Week. But the salmon that feed those bears at Brooks Camp are a different species than the ones that we observe on the coast. So they're sockeye salmon as opposed to chum and pink salmon. And they come from the Bristol Bay, which is on the west side of the peninsula, whereas the salmon that feed the bears that we studied come from the Gulf of Alaska on the east side. And as you can see here, the sockeye salmon abundance coming from Bristol Bay has increased over the years, and in fact has been record-breaking during our study. So if we had assumed that the coastal bears in Brooks Camp were experiencing, that the coastal bears were experiencing conditions similar to those of the bears in Brooks Camp, we would have been very wrong. So by looking at these multiple regions within the park and multiple resources within the park, we are beginning to see the links between the land and the sea and how they might affect bears and other species through time. but we still have a long way to go. I have a lot of people to thank for their help with this project, including mentors, partners within the park, pilots, collaborators, other graduate students. And so to all of you, thank you again. And I'll do one last plug to say that I'm now working for the Kodiak National Wildlife Refuge and the U.S. Fish and Wildlife Service in Kodiak. So if you have a desire to work in Kodiak and with bears, please reach out and get in touch. And with that, I'll take any questions.
Hi, my name is Tony Carnahan. I'm a graduate student at the Washington State University Bear Center. And today I'd like to talk to you about the use of triaxial accelerometers to identify grizzly bear behavior. We conducted the study as part of a larger energetic study that lasted the active season back in 2018. And our objective was to identify grizzly bear behavior using triaxial accelerometers. We were interested in basic behaviors, including walk, run, sit, stand, and lay, as well as trying to identify things like predation events. We had three trial periods that lasted May to June, July to August, and September to October. We used 7 bears, ranging in age from 3 to 16 years, and weighing from 90 to 265 kilograms. Each bear was fitted with activity sensors, primarily triaxial accelerometers, and they were given 24-hour access to a 10-hectare yard. We recorded behavior remotely using video cameras. The triaxial accelerometers we used were the Actigraph WGT3X, and they recorded at 30 hertz. We used these on all 7 bears, and they were easily traded out. The battery life on these only lasts about 3 weeks, which required that we trade them out and recharge them ever so often. These were deployed on both Telonics and Vectronics collars that the bears wore, and we also deployed 2 Vertex Plus Vectronics collars that recorded at 8 hertz. Like I said, we had to change the Actigraph triaxial accelerometers out, and so we use these aluminum boxes to encase them. They're waterproof, and they were easily attached to the collars. This is one of them on a Vectronics collar. Our bears are trained to go into a crate and are given diluted honey water, which allowed us to very easily trade out these sensors. To score the behavior, we used the Behavioral Observation Research Interactive Software, BORIS. And so far, we've scored about 80 hours of video using undergraduate volunteers and Bear Center staff. Like I said, we wanted to look at very basic behaviors, so we developed an ethogram that had locomotive behaviors, including lay, sit, stand, stand bipedal, walk, and run, and foraging behaviors that included grazing, digging, and feed on a carcass. We also had incidental behaviors like rubbing and other behaviors like playing, climbing. The bears have a large play structure that they can climb on in the yard. They enjoy hunting voles in the yard, so there's some instances of jumping or pouncing. And they also have access to a large pool that they can do various behaviors in. Some of these behaviors are hard to qualify, and not all times are the bears in sight. And so we also included an unknown or out of sight key. Since we did just want to, we were just interested in basic behaviors. The behaviors in the yellow box are the ones we concentrated on for the analysis, and the other behaviors were left out of both the training and testing data sets. Here's an example of one of the videos that we used. This is one of our bears grazing on a clover patch. When we look at the raw accelerometry data with the x-axis on top, y-axis on the middle, and z-axis on the bottom, we can visually see the differences between behaviors represented in the raw data. Now, to get the behaviors from this raw data, we calculated several variables, including static acceleration, which is a 3-second moving average of the rod axes. From this, we took the static acceleration and subtracted it from the raw data to get dynamic acceleration. And then we calculated vector dynamic body acceleration, or VEDBA, which is the square root of the sums of the squares of the dynamic accelerations for all three axes. And then we took a 3-second moving average of the VEDBA to calculate smooth VEDBA. We also calculated pitch and sway and partial dynamic body acceleration, which is the absolute value of dynamic acceleration for all three axes, for each of the three axes. We also looked at the ratio between VEDBA and each of the dynamic acceleration for each of the axes. To conduct the analysis, we took 1-second averages for all these variables, for each of these variables. We used package random forest in R and took 70% of the data for training and 30% of the data for testing. Each model was built using 500 trees, and we bootstrapped 100 times to find the best model. Our confusion matrix, to look at this, we're across the top, the columns represent the observed behavior. The rows represent the predicted behavior, and the bold numbers running diagonally are the agreement between the two. We had an out-of-bag error rate of about 12.5%, which we thought was pretty good for the data. When we look at the class errors, most of the larger errors come from underrepresented behaviors, including bipedal, standing bipedal, feeding on a carcass, digging, and rubbing. While the behaviors with the best error rates included like grazing and laying, which we had quite a few observations. And we had some that some of the observations, or some of the behaviors, there was the bears would, a lot of times when the bears would graze, they'd also be walking, and so you can see some errors associated with that. The variables that were most important in the analysis, according to the main Gini index, included the smooth VEDBA, partial dynamic body acceleration for all three axes, static acceleration for all the three axes, pitch and sway, as well as VEDBA. Moving forward, we're continuing to score videos, hoping to get some of the underrepresented behaviors. We're looking at how to handle mixed behaviors, i.e. walking while grazing, and then trying to improve the transitions between behaviors, because these are at 1 second intervals. Sometimes it's hard to really identify when one behavior ends and another starts. So overall, we're very happy with the models we've produced so far, and we'll continue to hopefully improve on them. I'd like to acknowledge several people, including Gordon Stenhouse from the Foothills Research Institute, Tim Lasky from the Medtronic Corporation, Frank Van Manen and Mark Haroldson from the Interagency Grizzly Bear Study Team with USGS, Sean Farley from the Alaska Department of Fish and Game, and our undergraduates Taylor Schaefer, Kaylee Wright, and Heather Havelock. Normally I'd be asking for questions, but hopefully I'll be able to answer some during the conference. Thank you for viewing.
Hello, my name is Cameron Mcclelland, and I will be presenting today on variations in grizzly bear habitat selection in relation to the availability of plant food resources. I'd like to thank my co-authors, Dr. Nicholas Koop, Ethan Berman, Dr. Sean Kearney, Dr. Cole Burton, Dr. Scott Nielsen, and Gordon Stenhouse. And our objective with this research was to measure when important grizzly bear plant foods became available for consumption using novel remote sensing techniques. This research is published in Applied Vegetation Science in 2019 and in Ecological Informatics in 2020. So first I'm going to go through how we created a fine scale regional phenology product, how we use this regional phenology product to infer plant food availability, and then how we determined variation in grizzly bear habitat selection based on shifting plant food availability. Our study area was the West Central Alberta area in Canada, so the Yellowhead Bear Management Area, which is heavily fragmented by resource extraction, forestry, mining, and sees substantial recreation use in the form of OHVs, hiking, hunting, fishing, etc. So how do we measure phenology? Well, we did this at two levels. We first use time-lapse cameras and field observation. That's our plot level. From this, we're able to determine phenology dates, such as green up, maturity, and senescence, as you can see in these pictures. We're also able to determine when plant food species become available for consumption. So in this case, blueberries are able to be consumed when they're blue and nice and juicy. We're also able to extract greenness indices from a time series of time-lapse images. So this will give us the start and end of season for a site, and will also give us the exact date of seasonal max. However, this is difficult to scale to a regional level, and for this reason, we need remote sensing with satellites. In this research, we use two common remote sensing platforms. The first is MODIS, which has a 500-meter resolution, a daily return, and has data from 1999 till present. Next, we use Landsat, which has a 30-meter resolution, so much finer than MODIS, has a 16-day return, which can be longer with cloud cover. So this is also longer than MODIS. And it has data from 1984 to present. And what we wanted to create was a daily phenology product with a 30-meter resolution. To do this, we use data fusion methods between MODIS and Landsat, which incorporates the daily temporal resolution of MODIS, so the fine scale temporal resolution of MODIS, and the 30-meter spatial resolution of Landsat. And that is the best of both worlds. We've entitled this product DRIVE, which stands for the Daily Remote Inference of Vegetation. Here is DRIVE in action. So March into April, we start to get a little bit of green up. Coming into May here, huge green up across our study area. Into June, green up is still persistent, a little bit more, maybe into July. And we see seasonal max, and then we see senescence begin in August into September, October, and finishing in November. And this next bit is just to show you at which scale we did this at. So the Yellowhead Bear Management Area where we did this is a very large area, around 30,000 kilometers squared. And we were able to create this product over the entire area. And one thing that we found that was interesting with this is that start of growing season is shifting, probably likely under changing climatic conditions. And we analyzed this across all elevation classes and land cover classes from 2000 to 2018. And we found significant change amongst almost all elevation and land cover classes. We also found that start of season was coming a maximum of 0.78 days earlier per year. And you can even see this visually in this figure over here, where in 2010, much browner. So that means that start of season was later, and compared to 2018, where it's almost all green, where start of season is much earlier. So next, we want to use DRIVE to create regional plant food availability layers. To do this, we looked at 8 plant food species: alpine sweet vetch, which is a root species; dandelion, a forb species; clover, another forb species; horsetail, forb; cow parsnip, a big forb species; buffalo berry, important berry species; huckleberry, also an important berry species; and velvet leaf blueberry. And these are ordered in descending order of later availability. And for each of these species, we created annual food species distribution models. To do this, we use maximum entropy models, which is a computer learning algorithm and only requires presence data. We created these models from 2000 to 2018. As you can see, we use climate variables, terrain variables, but most importantly, we use annual variables. These are the variables that change annually, which consist of land cover class, distance to forest edge, and percent canopy cover. And here's this in excuse me. So this is buffalo berry presence from 2000 to 2018. We see, most importantly, that when there's cut blocks, there's less presence, but as the cut blocks grow in, the presence becomes more abundant. So next, we want to create daily availability layers. And here we're using velvet leaf blueberry as an example. So first, we determine the day of the year in which the start of availability begins. So this is for blueberries, when the blueberries are blue and juicy and edible, and when it ends, which is when the berries go to seed. However, to extrapolate this to from site level to regional level, we need to use our drive curves. So we extract a drive curve for each of our ground sites. We then split the curves into 2 halves. So 0 to 100% is the green up portion of the curve, and 100 to 200% is the senescence portion of the curve. And we use percentages to allow start and end of availability dates to vary by land cover type and by elevation, essentially by how phenology curves shift across the landscape. We then extract percentages along this curve that correspond to our the start of food availability and end of food availability dates. This creates our food availability window. We then relate these 2 percentages. The start availability begins at day of the year 212. This relates to percentage 123, and our end of food availability is the day of the year 254, which corresponds to percentage 160. Once we have these percentages and have found an average percentage across all our sites, we're able to extrapolate this over the entire landscape using our drive product. We then mass this using probability currents above 80% from our species distribution models. And this is our final product. So this is cow parsnip availability in the year 2017. So as we hit June here, you can see that all of a sudden cow parsnip becomes available for consumption. Its stalks are nice and green and leafy. But as we get to the end of July, it quickly turns into woody stalks and becomes less available for consumption across the landscape. So how does annually varying plant food availability affect habitat selection? So to do this, we model selection for available foods. We use GPS collar data collected by FRI, and we created seasonal resource selection functions across 3 years. We did this in a wet year, a dry year, and an average year, and we used generalized linear mixed effects models. We compared 5 models using AIC and average correlation of probability bins, and we found that our annual availability model performed the best compared to all our other 4 models. We then created species-specific models for wet, dry, and average years. Briefly, here are some results that we found. So we found that in the dry spring, selection for roots was stronger and occurred earlier. In the wet summer, the length of selection increased for forb species. And in the dry fall, the period of selection was longer than in the wet year for berry species. However, in the average and wet year, selection for berry species was stronger. So what does this mean for management? With these techniques, managers can determine how shifting phenology affects food sources. And from this, we may be able to predict where grizzly bears will be on the landscape based on differences in food availability. So for a wet year versus a dry year versus an average year. With this, we may also be able to develop dynamic Arctos plans that may help to mitigate human bear encounters during these different years. And as a take-home message, what I really want to emphasize is that these methods and resulting product can provide a high level of detail into the shifting, how plant availability is shifting under climate change, and that grizzly bears select for different habitat based on annual food availability. So I'd like to thank everyone for listening to this presentation. Again, my name is Cameron Mcclelland. I'd also like to thank all our partners, and especially the Integrated Remote Sensing Studio. Thank you. If you have any questions, please ask them.
Hello, everyone. My name is Justin Tiesberg. I'm a biologist with the United States Fish and Wildlife Service Grizzly Bear Recovery Program out of Libby, Montana. Our focus in the office is on monitoring recovery of 2 populations of grizzly bears, the Cabinet-Yaak and the Selkirk. So a little bit of geography. This is where the Cabinet-Yaak and Selkirk ecosystems fall. These are the recovery zones for both. They're really 2 separate populations, and they're separate from the NCDE to the east and the Yellowstone to the southeast. We're dealing with 2 small populations. In the case of the Cabinet-Yaak, about 50 to 60 bears. In the case of the Selkirks, maybe 70 to 90 bears. Both populations suffer from top-down limitation in the form of human-caused mortality. Both populations, in a relative sense, also have much lower amounts of habitat available to them when compared to the Yellowstone or the Northern Continental Divide. Their diet is also different in that it's more composed of plant matter, so green vegetation, berries in the midsummer to early fall, some root digging available to them, but on average, very much less on the animal matter available to bears in the Cabinet-Yaak and Selkirks. So we asked the question, really, are there any bottom-up limitations to the population when we look at foods available to bears and habitat quality? And we took on this overarching question, trying to look at food items used through stable isotope analysis. It allowed, very briefly, isotope methods. I'll talk around isotope ratios of nitrogen in this talk. And what we're tracking is the ratio of a heavy nitrogen isotope relative to the light atom of nitrogen. And that ratio increases with trophic level. The 15 atomic weight is retained at a higher rate than the 14 atomic weight in the body of a consumer. Cabinet-Yaak Selkirk captured events. We collected hair samples and blood samples. And where possible, we separated the hair sample into two components. The guard hair sample, which is the longer hair shaft, which grows during the summer months, and the underfur sample, which initiates growth in early fall, and really is that insulative underfur sample that keeps them warm in the den. They grow at two different times, and so they're cataloging the chemistry of the bear at two different times. So it gives us insight into what the bear was doing as far as its food economy at two different time slices. We also took blood samples, which can get you even tighter period of time as far as their dietary insight. So bears, as an omnivore, can exist anywhere from 100% herbivore to 100% carnivore. And so that's the question exactly. Where on that spectrum do Cabinet-Yaak Selkirk bears fall? Okay, so what did we find? Focus on Cabinet-Yaak bears here first. These are guard hair samples, so indicative of that summer signature. And we get a kind of a more precise understanding of what's going on out there. Roughly 90% of their diet in the summer months, the average grizzly bear in the Cabinet-Yaak, is composed of plant matter. You can see males use a little bit more, significantly so, than relative to adult females and subadults. But the story here is that there's very little animal matter used in the summer months by Cabinet-Yaak bears. The same is true of Selkirk bears. Adult males even less so in the Selkirks as relative to the Cabinet-Yaak. So both ecosystems, the story is the same. They exist largely as primary consumers during the summer months. And those values relative to other ecosystems, you can see they're on the lower end as far as the trophic spectrum there. Much more so than the east front of the Northern Continental Divide and the Yellowstone. Maybe similar in what they're doing relative to trophic level with the northwest part of the NCDE. So really, that's the summer. It's marked by very little animal matter in the diet and a lot of vegetation. But we had this underfur sample where we could look at any shifts that might occur as we move into the fall. And what we found is that they do, as a percentage of their diet, double the amount of animal matter in their diet as you move from summer to fall. Adult males might even triple what they're doing as far as their consumption of animal matter, again, as a percentage of their overall diet. We figure that this is influenced by hunting season, so wounding loss and viscera or gut piles that are available to bears in the fall season during hunting season kind of influence this increase in the use of animal matter. We also asked the question whether we can figure anything out as far as other foods used through isotope methods. This is pretty standard procedure just to make sure that all of your food sources are unique in their isotope ratios. We collected other foods across the Cabinet-Yaak Selkirk, including berries, which are a more important food, we figure, to Cabinet-Yaak Selkirk bears, primarily in the form of huckleberries. But so we collected, processed, analyzed samples. of multiple berry species across the Cabinet-Yaak and Selkirk. Pretty good geographic breadth here across the landscape. Fairly decent representation of most species, most definitely huckleberry. But mountain ash, we were a little limited in the sample size, and I'll sort of show you the outcome of that here in the next slide. So isotope ratios, we're looking at nitrogen only here, of these berry species relative to all the other vegetation collected in our sampling. Other vegetation fell out pretty tight. That's all the way on the right there. These are confidence intervals, 95% confidence intervals around the mean. Huckleberry, uniquely low relative to serviceberry, buffaloberry, and other vegetation. Mountain ash, not quite sure what's going on there, overlapping with serviceberry and huckleberry. I think our sample size was pretty limited. We're trying to tighten that up a bit and see if it has a similar isotopic signature with huckleberry. But long story short here, huckleberry shows some promise in that we might be able to track its use through isotope means as well. So in the next couple of slides, I'm going to show how this low berry value actually fits the modeling process a little better than what we understood before. So this is a representation of the mixing space of different dietary endpoints in an isotopic modeling sense. So on the x-axis, we have carbon isotope ratios. On the y-axis, we got nitrogen isotope ratios. The circles are bear values, tissue values, isotope ratios. And the endpoints in this triangle are representations of what the bear's tissue would look like isotopically if they fed on 100% vegetation, 100% meat, or 100% anthro, which I'm calling human foods, right? Trash, which is typically higher on the trophic level, and more, it contains more corn, so lower in its carbon isotope ratio. And you can see that a lot of the bear values fall outside of the polygon, which doesn't make too much sense. But if we this low berry value, low on the nitrogen scale, now the clear majority of points fall within the polygon. So really the modeling only makes sense if we include berries in the model. So we went forward and modeled the guard hair values for adult females using this 4 endpoint model. And we found that upward, on average, of a quarter of the assimilated carbon and nitrogen for adult females comes from this berry source, upwards of 44% for some females. This is a significant amount of the assimilated carbon and nitrogen, especially if you consider that guard hair initiates its growth in the late spring or so, well before berry season starts on July 15. Berry season is typically July 15th through September 15th in the Cabinet-Yaak Selkirk. So we're able to account for an important food source and the percent of the diet composed of it. So all this together, what does it mean? We know that they use berries at a pretty high clip in the summer and fall. We know that protein might become more available in the form of animal matter in the fall time. Do we see any indication of bottom-up limitation in these populations? Well, there's no indirect evidence, and one example is body fat contents that we collect at capture of adult females. And by the time fall rolls around, the average female is above a threshold, 20-21% body fat content, that indicates high likelihood that the female is going to produce cubs the next year. On top of that, also, reproductive rates in the Cabinet-Yaak and the Selkirks do not significantly differ from other ecosystems. And so there's no indirect evidence. I do want to dig into whether there's direct evidence of whether transition probabilities are explained by berry consumption, level of berry consumption, or trophic level. Higher trophic level, more chance that a bear is going to transition from being solitary to with cubs. So with that, I wanted to thank everyone. And if there are any questions, please tune into a live Q&A on September 22nd. Thanks.
Hi, everyone. My name is Hertel. A lot of you may know me. Thanks for giving me the opportunity to give this presentation in the behavior session. And my talk will be about beyond population means, individual behavioral and dietary specialization in Scandinavian brown bears. Here you can see two female brown bears. They have the same age. They were recorded in the same year. They have a fairly similar habitat composition, and yet they seem to show very different behavior. So the red one on the right-hand side has a much smaller home range. It has shorter daily movement distances. Meanwhile, the one on the left-hand side, the blue one, has a home range size is approximately 3 times as big, with much longer daily distances. And here's already where my research interests really lie. So why do individuals, and for example, these two, differ in their space use, and how stable are such differences over time? There's, of course, a lot of factors that can contribute to this variation and can be the driving factor. For example, differences in sex, females and males, differences in reproductive status, the hunting season, animals shift their behavior when there's all of a sudden mortality risk on the landscape, differences in habitat composition, or in food availability. Yet beyond these driving factors, so to say, the question is, do we still see differences, and why is that the case? So if we want to quantify such differences after controlling for things like sex and reproductive status and season, what we need is repeated measures of behavior. Movement data are particularly suitable to do that. So kind of borrowing techniques from the behavioral ecology literature, what I have been doing is I've been repeatedly quantifying behavior for movement data, and then I've quantified whether individuals differ in their average response. So in this case, for example, we have 4 toy individuals that were measured in 4 time steps. And these individuals seem to display consistent variation in their behavior, which leads to differences in average behavior for any kind of behavior. A second metric that I'm using, that I'm going to present to you today, is a metric called predictability, and that is more looking on the within individual variation and quantifying the standard deviation of behavior around the average. So as you can see here, individual 1 and 3, for example, have a high standard deviation around their mean. Meanwhile, individual 2 and 4 seem to be fairly persistent. And if we're controlling for outward factors that can contribute to this variation, then there's still biologically relevant information here that some individuals may just be a lot more predictable than others. The study system I'm working with is the Scandinavian Brown Bear Research Project. And it's extremely suitable to do that because it has like these long time series of individual data where we have movement data, we have body mass, we have life history trait, we have hair samples for individuals, sometimes ideally over their entire lifespan, and we have a fairly complete pedigree of the population. And these factors allow me to control for a lot of things that may actually lead to these among individual differences. So the 2 topics I would like to present to you today are A, behavioral specialization in movement and dial activity, and B, behavioral specialization in diet, so dietary specialization. So what we did for the first part is we used movement data from GPS collars and activity data from acceleration centers in the GPS collar. And we quantified 2 metrics. First of all, we looked at dial activity, so daily diurnal activity. So we looked at the 24 period, We took the acceleration data and we looked at how much activity would be placed into the daylight hours of the day. And here in this graph, it would be during the yellow hours of the day versus the night hours. And the index is controlling for day length, and it ranges from -1 to 1, -1 being individual that places all its activity during the night, so strictly nocturnal, and 1 being an individual that is strictly diurnal, so having all the activity during the daylight hours. The second behavior that I looked at was daily movement distances. So I quantified for every day the cumulative kilometers walked by the individual given a 1-hour location date. And for the whole study, I used 62 female bears, individuals, monitored in 187 years, and in each year monitored for 50 days. So that gave me a huge sample size to draw means and standard deviations around means. So first of all, the mean behavior, so it's personality. So what we found is that, for example, for daily activities or diurnal activity, some individuals in our population here in white and in yellow on the left-hand side of the graph are fairly nocturnal. So most of the activity is placed during the night hours. Meanwhile, other individuals in red on the right-hand side of the graph are fairly diurnal. So most of the activity is placed during the daylight hours of the day. We also found, less pronounced so, however, that some individuals consistently over the 50 years in a day and over many years walk over shorter distances, whereas other individuals walk over longer distances per day. And interestingly, these two behaviors, so these average behaviors being either more nocturnal or diurnal, or being someone that is more sedentary or very moving, these correlated with how predictable individuals were in their behavior. So for example, for daily activity, those individuals that were nocturnal here on the x-axis on the left-hand side, they were also fairly predictable in their behavior. So over 50 days within a year, for example, they would always be nocturnal, as nocturnal as they could. Meanwhile, those individuals that were more day active, they were also more variable in their behavior, and were sometimes day active and sometimes were night active. And the same pattern was found for daily movement distance, where bears that walk over shorter distances are predictable in that and very consistent. Meanwhile, bears that walk over longer distances on average have a higher standard deviation around this mean. So in essence, we have some bears that are night active, and they're sedentary and they're very predictable. They always display these behaviors. Meanwhile, others are day active, mobile, but unpredictable in their behavior. And we're not really sure why this is the case. So we have two possible explanations. So either the risky bears that are day active and moving switch their behavior in reaction to actually meeting someone, because being day active and being active also increases the likelihood of meeting people in the forest. The alternative explanation is that unpredictable behavior can be an adaptive strategy to avoid predators, which maybe for humans actually doesn't work that well. But in other examples of predators and prey, being unpredictable is a strategy to confuse the predator and reduce predation risk. Now, the second part of my presentation, I would like to spend on dietary specialization. And to shortly summarize what I mean is, if we have a population niche of dietary items that can be consumed, we can either have a scenario where all individuals in the population are generalists and kind of feed on the full spectrum of diet items of the population, or we have individual niche specialization, where some individuals really concentrate on certain items in the diet of the population. And for brown bears, this could, for example, be measured by looking at how carnivorous they are. Maybe some individuals in the population rather feed on grasses, fruits, berries, etc., and others concentrate more on eating animal matter. So for Scandinavian brown bears, we have as for all brown bear populations, a wide area of dietary items, for example, grasses, moose calves, ants, berries, and carcasses. So carcasses in the forest, slaughter remains, for example. And because the project takes a sample, a hair sample, every time a bear is captured, we can measure their stable isotopes in the sample, which encode approximately for the dietary niche in the preceding year when the hair was growing. We were able to collect 500 hair samples from 108 females and 92 male bears. And we quantified the trophic position from the delta 15N signature, which is an indicator for how carnivorous a bear is relative to other individuals in the population. So our first model looked at, do we actually have individual variation in this trophic position? And the second one looked at, what are the determinants of such individual variation? So what we found was, first of all, that males, especially large males, so beyond 150 kilograms of body mass, are a lot more carnivorous than females are in general. It's probably because they can monopolize carcasses, and they may also prey on neonat bear cubs. But second, beyond that, we still found that there's strong individual variation in the trophic position. So individuals that are on the right-hand side of the graph are more carnivorous than individuals on the left-hand side of the graph. The big line indicates the population niche for females and males separately, and the smaller lines and dots are representative of the niche of one individual. So what determines whether a bear would be more or less carnivorous? We were in particular interested in looking at relatedness, so heritability, spatial similarity, and maternal learning. And what we found was that the trophic position of a bear was determined by the trophic position of its mother, however, only in the first years after the family separated. So to make this more clear, in year 1 and 2, after separation, the offspring's position resembled the mother's position, but 5 years after separation, the position was fairly different, indicating that there's individual learning kicking in after a while. Additionally to that, spatial proximity also played a role. So individuals that lived close in space to each other were more similar in the trophic position, indicating that there might be variation, for example, either in moose density or in hunters hunting moose and leaving carcass remains in the forest. So to sum this up, we see that there is extreme value in long-term monitoring data when we want to reveal individual specialization in behavior or diet, movement, or activity. We also find that individuals that show, especially females that show a risky behavior, seem to be less predictable, and we don't know exactly why, whether this is actually an adaptive response or whether it's just a byproduct of meeting people in the forest more often if you're more risky. Initial analyses, by the way, did not reveal any survival benefits of this unpredictable behavior. And there seems to be strong individual variation from a more vegetarian to a more carnivorous diet in individuals where males in generally are more carnivorous than females, and diet selection is initially earned from the mother. Well, I hope you enjoyed this talk and you have lots of questions. And if you are curious to quantify these measures in your own population, please contact me. I'd be happy to help you. Thanks to my collaborators, in particular, Neil Stingemanse, Thomas Müller, and Andreas Cedresa.
Hello, everyone. My name is Aida Paris, and today we'll talk about conference patterns, how they change over time and space, and how anthropogenic disturbance may disrupt these patterns. Most animals move, and movement is a tool that animals use to satisfy their biological needs, such as searching for food, shelter, through the interaction with the environment. This movement takes place in a particular space, which is commonly recognized as home range. Home range has been described in many different ways, but I find this definition as the most accurate one. Here, the home range is defined as dynamic patterns of the space use determined by complex interactions between individuals and their environment. Let's pay attention to this concept of dynamic patterns of the space use. This definition is already considering that the environment fluctuates. If it changes, the interaction with it will also change. So we may expect that an individual will be constantly readjusting the home range over time. To better understanding, let's move forward. So for example, an individual moves, interacts with the landscape and uses the area that we will consider the home range, but suddenly decides to move away from this home range area and makes an exploration, followed by the establishment of another home range. And then again, makes another exploration, and again establishes another home range. Home range estimation is not an easy task, especially if we want to estimate or to capture this dynamism. First of all, home range integrates different components, not only the size, but also the location, the shape, and all these components, they represent the space use of an animal. The home range dynamic is affected by many different factors. For example, internal and biological factors like sex, age, or internal aspects of individuals, they may affect the way the animal moves. And this movement may affect the way the home range changes. These biological factors are directly related with the biological needs of animals. So these biological needs, they occur at different scales. So for example, foraging or resting, they are activity that they are routinarily and they occur at daily scale. However, there are other activities or biological events like mating or hibernating, which scale is longer. So to consider an appropriate representation and estimation of the home range, we may consider the temporal scale that will follow biological meaningful times, like circadian, breeding cycles, and the total lifespan. External factors are also crucial in the home range dynamics. Food availability in the environment is not uniform, and as food resources change in time and space, the home range of individuals will also change. Then also the expansion of human activity has been shown that disrupts natural behavioral patterns of animals. For example, the increase of human infrastructures or outdoor activity may create physical movement barriers for individuals or trigger human avoidance strategies, which may also influence the home range dynamics. So the purpose of this study was to describe the dynamics of the home range patterns in the brown bear of the northern Carpathian population. And we focused on 3 research questions. Is the home range dynamic? Is there a seasonal pattern in the dynamics of the home range? And is the dynamic pattern of the home range affected by anthropogenic disturbance? Here we used 29 bears monitored with GPS collars from 2013 to 2020 from the Northern Carpathian population. Then we estimated the home ranges using the local convex hull estimator, and we used a daily scale. We wanted to use 2 different components of the home range to characterize the dynamics of the home range patterns. So we used the daily home range size as a measure of the minimum area needed per individual per day, and the shifting distance as a measure of changing in the location between consecutive daily home ranges. Then to explore the dynamics of the home range, we first modeled the relationship between the daily home range size and the shifting distance with a generalized linear mixed model. We include the sex as a fixed factor and the bear identificator as the random factor to control for repeated observations per individual. Then to explore the temporal patterns of the seasonal patterns of the dynamics of the home range, we used a general additive mix model. We again included the sex as a fixed factor and the bear identificator as a random factor. And to account for the seasonal pattern of the home range components, we included a smooth of the cyclic effect of the day of the year. We considered here separately two different models, first for the home range size and then for the home range shifting distance. Then, finally, to assess the effect of human-related variables on the dynamics of the home range, we considered the presence of human infrastructures and the outdoor activity as hiking traits. So the presence within the home range. Then also we built two different models, one for the home range size and the other one for the shifting distance, by using a general additive mix model. So what we got so far is the home range dynamic. We observed that all 29 bears tracked in our population showed dynamism. First, on average, bears used an area of 0.7 square kilometers per day and moved their home range location from the previous day 2 kilometers per day. And then second, we found a significant positive correlation between the size and the shifting distance of the home range, which already shows that there is a dynamic pattern in this home range. And this indicates that when bears used larger area, the distance of the change of the location increased. Then we also discovered that the home range pattern has a seasonal pattern actually. We observed that the size and the shifting distance of Northern Carpathian brown bears generally showed the minimum values at the beginning and at the end of the bear year, considering that when the individual gets out of the hibernation period and enters in the hibernation period. So the minimum values, they are at the beginning and at the end of the year, and the maximum values, they are in June, coinciding with the summer solstice. Generally, as we can observe in the graphs, we can see that females, that they are on black, showed more pronounced seasonal pattern than males, that they are on orange. Then, considering if anthropogenic disturbance affects the dynamics of the home range patterns, we can say yes. In areas with human disturbance, as we can see in the graph, in the column where it says yes, we can observe that for both human infrastructures and outdoor activities separately, brown bears distorted the seasonal pattern and flattened the line. So only when the home range of bears contain areas without human disturbance, you can see it in the column where it says no, show that clear seasonal pattern. We also observed that for the daily home range, the females and males responded differently to the effect of human disturbance. We can see in the column where it says yes, that females, they preserved the seasonality, but they flattened the pattern, while males, they lost completely the seasonality. The model describing the effect of human infrastructures explained 43%, and the one of outdoor activity, 35%. For the daily shifting distance, we found also a similar pattern, but the models explain 31% for the human infrastructures and 27% for outdoor activity. However, if we observe the graphs, we can see how females and males, they responded identically. So both females and males, they lost completely the seasonal pattern. So to conclude, we can say that brown bears used the space use in a dynamic way, that both the daily home range size and shifting distance can be used to describe the dynamism of the spatial use. The daily home range components, both they follow the seasonal pattern, and that human disturbance affects the seasonality of the spatial use of bears, disrupting the seasonal patterns. Thank you very much, and I hope you enjoyed the presentation.